Let's start. Assalamu alaikum warahmatullahi wabarakatuh and good evening. So, as you all know, this is a part of the seminar series conducted by the Muslim Majlis of University of Sijavadanpura. So, today I am hoping to do a revision, osmoregulation, and excretion. Uh, First of all, now I know you all will be so thrilled because you might be the next batch to receive the result. And you might be thinking how are you going to face your exams, so don't get tensed or nervous. Just go with the flow and try to revise everything you studied. So today um, we will do a revision on osmoregulation and excretion. So you can interrupt me anytime. You can turn on your mic and ask me any doubts, or if you have any confusions, if you think something is not clear, you can really turn on your mic and ask me, because if you message me, maybe I won't be able to look at your messages. So please turn on your mic and ask your doubts. Also, you can answer the questions I asked. Mm, Okay, so the reason for me to choose osmoregulation and excretion for you for the revision today is because it is a very easy topic among all the topics in your unit 5. In animal form and function, there are so many subdivisions you might be studying. uh, I felt small regulation and excretion is the most easiest part. Also, you can score a lot. And also, when I was doing my A levels, I expected osmoregulation and excretion in essay. So I was hoping for that essay to come after all the researches I've done with the past papers. So I thought this will come, but unfortunately, it didn't show up. So even last year, 2022, also there was no any essay questions on osmoregulation excretion. Maybe we can expect this time. So this is a very easy topic to score. So therefore, don't avoid this topic. Try to get the idea uh, as much as you can and try to remember it rather than wasting your time on just reading up the notes. Okay, so now we will go into osmoregulation or excretion. So what is the first of all, we need to know what is the importance of osmoregulation excretion, right? So these are the topics we will be considering today. There are so many other topics that once again. Okay. So these are the subtopics of osmoregulation and excretion we will be seeing today. The importance of nitrate and excreted nitrogenous excretory products and the living environment, the relationship in the living environment. Then excretory structures of animals, I mean the diversity, human urinary system, the process of urine formation, and the role of hormone. So what I missed here is the diseases and disorders related to osmoregulation excretion, because that is a very important topic and you can just go through it and understand. So we will be focusing on these topics. First of all, okay, what is osmoregulation and why are we studying excretion together with the osmoregulation? That is one of the questions we might be having. Osmoregulation and excretion are connected to each other. We will see how later. So first we need to know what is osmoregulation. So it is a process. It is a biological process. As you've been studying from the beginning of the unit 5, you know it is a process. It's a biological process that is happening in the living body, not only humans, any the other animal body too. So we know in our body we can in our body there are cells. Around the cells we have some interstitial area. Then we have the blood vessels and we have blood. So this is our internal structure of the body. So there are solutes in the body, in our body. Okay, within the body we have different types of solutes. They are in certain concentrations. So these concentrations should be maintained in a particular range. So wherever you study about the biological process, you won't be studying anything very specific. There's no anything like very exact. Rather, you will be having a range. Take temperature, you have a range. If you take blood pressure, you have a range. Glucose level, you have a range. So you have a specific range. So you have to maintain this solute concentration, the concentration of the solutes in a particular range, within a range. Also, our body is made up of water. So there should be water, or there should be some range of water. So we have to maintain our body within that range. So here, osmoregulation is the process to control the solute concentration and the water balance within our body. So that is the target of osmoregulation. So you might have studied amoeba, paramecium, where in unit 3. So I underlined. So you might have studied them. They are the unicellular organisms studied under kingdom protista, I think so. So that is in unit 3. You might have studied, and you might have come across a word called contractile vacuoles. So these are the structures that are used by the unicellular organisms such as amoeba, paramecium. So they are osmoregulation. So they are also living body, right? They are also living structures, the living body. So they also have solute concentration and water in the body. They also have to regulate the osmolality or the osmolarity in their body. So they have specific structures called contractile vacuoles. There's another vacuoles. So this is contractile vacuole. But the animals, I mean the animals mean except those animals which come under the kingdom Animalia. So they have developed different structures for osmoregulation. They have different structures. So we will, when we go through the excretories, when we go through the diversity, we can understand what are the different structures they have for osmoregulation. Okay, so this is the need of osmoregulation. So what is the summary of osmoregulation is you have a certain concentration of solute and water in your body. You have to maintain them within a particular range. That is the target of osmoregulation. Then you have anemia, paramecium, which have contractile vacuoles for the osmoregulation. Now, okay, now what is excretion? Excretion means from your small age itself, you might have studied that there are so many chemical reactions that are taking place in our body, right? So those chemical reactions, what is the very famous chemical reaction that you have studied from grade 6 itself? You might have studied the equation for the chemical reaction, biological process from grade 6 itself. You have studied. So can you say the famous one? Sorry, I couldn't hear. Yes, respiration. Okay, respiration. Yes, respiration is the very famous uh, equation. Okay, you have studied that from grade 6 itself. From the first unit itself, you have studied the respiration. So what has happened? So your glucose, the food you take glucose, will be oxidized by oxygen, and it will give you carbon dioxide and water with energy. So this energy you will use for various process in your body. So here, this carbon dioxide is a waste product that is produced in our body. So not only carbon dioxide, there are so many other things. So you can study them in basics, right? So after that, you might have studied. We will also study later. Okay. So carbon dioxide is one of the waste products. So there are so many waste products that are produced in our body during this chemical reaction. Okay, so these waste products, so as the name itself says, waste should go out. So they are toxic. When it is accumulated in our body, it is toxic to our body. So we have to dispose in some way. So for that, we have a special biological process in our body, excretion. That is excretion. So excretion is the removal of nitrogenous metabolites. So what do you mean by nitrogenous metabolites? So this comes under the other metabolite metabolic base. So nitrogenous metabolite, the very famous one is the ammonia. So you can get ammonia from proteins, uh, proteins as well as nucleic acids because they have nitrogen in them. We will see later also. So removal of these nitrogenous metabolites and other metabolic waste products in our body from the body is called the excretion. So this is why we need excretion. So what is the famous one we all know, like we say is is it a waste product? So we often call it as waste product, but metabolic waste, it is not a metabolic waste. Rather, it's just the undigested food that we are removing from our body. But you know, the food will be mixed with bile. So bile has some pigments and they are waste. They are also removed with the feces, but we don't call feces as a waste product because it is not a metabolic product. It's just undigested food. Okay, so that is a very famous question that is asked in structure. In the structure question, they might ask why is defecation is not considered as excretion. So you have to give the reason why. So now this is the need of osmoregulation. So what is osmoregulation and what is excretion? So why we are studying together is because those systems which carry out osmoregulation excretion are connected to each other structurally as well as functionally. That is the primary reason why we are studying them together. Okay, so I hope you got a clear idea about osmoregulation excretion. So what are the questions you might be asked is they might ask what is osmoregulation or what is excretion and sorry. And the famous question, why is purification or considered as excretion? Okay. So now the importance and then so we have to know why osmoregulation, excretion are important. So I just normally explained you a little bit before, but we will see more deeper here. So whenever we study any biological process, we need to know why. So what is the important and why do we need that? So when, from the beginning itself, when you are studying the unit 5, you have gone through uh, what, so many processes. For example, digestion, circulation, uh, respiration. So those, you know, are the systems related to those biological process. So first thing you studied is what is the need of it? What is the importance of it? Why it is there? That is what we are going to study here. So I have tried to make a simple like summary of the importance and need of personal regulation and the excretion. If you want, you can note. It's not necessary. So, what is the main aim of our body? It is for the effective body function. All the process in our body target two things in our body. They are the effective body function and the survival of us. Okay, those two are the very important things that are those two are the objectives of all the biological processes in our body. For that, we have to maintain constant internal environment. Like I told you earlier also, like we have to maintain the concentration of water or solute within a particular range. That is the favorable limit in our body. So for that, we have by that we can maintain a constant internal environment. So what is internal environment? What do you mean by internal environment? So internal environment. So internal environment is the part. It is the area out of our cell. So if I take this is one cell and this is another cell. The area between the cells are the internal environment. Okay, this is the internal environment. So internal environment is not the area inside the cells. No, it is not. Okay, internal environment is the area outside around the cells. Okay, just remember that this internal environment can directly impact on the cells, and uh, very minimal change in the internal environment can drastically change the process that is happening in the cell. So that is why we have to maintain a constant internal environment. Okay, so we have to maintain we have to maintain this internal environment in a constant, the relative concentration of water and solids. So we know the animals, they regulate the chemical composition of their body by balancing the uptake and loss of water and solids. They can uptake the water and they can lose the water. Also solids, they can do that. So that's what I've written. So to maintain a constant internal environment, there is uptake of water and water and solids, as well as loss of water and solids. But when there is excessive uptake, so this you might have studied in unit 4 also, when you studied about the concentration of the solutes and all. When there is excessive uptake, what happens? The cells will swell and they will burst. And if there is high loss of water and solute, the cell will shrink and it will die. So this you already studied this because uh, you studied in plant form and function under particular unit, so you know this way. So here we need osmoregulation, that we are not letting the cells to swell or to shrink. Okay, so here also the driving force of loss of water in plant is just like in animals, just like the plants, is the concentration gradient. So when the concentration gradient is there, from the high concentration to low concentration, the solutes can move across the cell membrane. So this is why we need osmoregulation. This is the reason why we need osmoregulation. So animals have evolved different osmoregulatory strategies based on their environment. So environment plays a major role in determining the osmoregulatory strategies. That you have to remember well, because we will study that again in two parts. We will see. So that is very important. So this is why we need osmoregulation. So next, it is excretion. So there are so many chemical reactions that are taking place in our body. So we call them metabolism, and so many toxic products are produced by metabolism. So we have to safeguard our internal environment, safeguard the composition of the internal environment by getting rid of all these toxic products. So otherwise, what will happen if the excretory end products and those waste products, they are the same. Excretory end products will become toxic to the body cell if they are accumulated in the body. So I already told us. So this is proteins. Okay, we take food. When we take food, we are taking glucose, proteins, what else? Carbohydrates, proteins, the lipids, nucleic acids, everything. We are taking that is our nutrition. So proteins, they will be broken down within the body cells during the metabolism, and the amine group in the amino acid, that will be converted to ammonia. So I hope you can remember the structure of an amino acid. So here, this is the amine group. This will be removed and converted to NH3, which is a toxic product to our body. Okay, this is one of the main toxic products to our body. So here it is. And then glucose, you already know that is because of the respiration. Respiration, and the carbon dioxide will be produced during respiration, and it should be excreted. Okay, so these are the main products that will be formed by the metabolism. So we have to release them out. If we don't release them out, they will be toxic to our body. If those toxic products are not released released out of our body, it will be toxic to the body cells. Also, we know the carbon dioxide that is released during the oxidation of glucose, It is a weak acid. You might have studied in chemistry. So it is a weak acid. Also, this ammonia is a weak base. So now we have weak acids and bases in our body, and they can alter the acid base balance in our body. They can alter the acid base balance in our body. So this acid base balance, when there is an imbalance in this balance, it will lead to so many consequences in our body. The very famous popular example, is the denaturation of proteins. This is studied in unit 2. So you might have studied high temperature, acid bases, some detergents. They can denature the proteins. They can denature the protein. That is, the 3D structure of the protein is lost, and it is no longer can do its normal functions. So what is the negative effect of this? Most of the enzymes in our body are proteins. Also, so many other things are made up of proteins. So those things will be altered by this acid base balance. So this is one of the very main reason why all the excretory products should be removed from our body. So we have to remove the excretory products from the body, so therefore we can maintain the internal environment with favorable conditions for our, what is the ultimate objective of all this process: effective body functioning and survival. All right, so that is the need and importance and need of this osmoregulation and excretion. So we are moving into the second part. So that is relationship between the metabolic substrates and excretory products. So what are the metabolic substrates? You can really answer.
Carbohydrates, fats, lipids, nucleic acid, and proteins.
Exactly. Okay. So those are the metabolic substrates that we are taking through our nutrition. Okay, we are taking into our body through nutrition: carbohydrates, fats, proteins, nucleic acids, and they will go on to go under some metabolic process, and they will give us so many good things to our body, as well as waste products to our body. So the excretory products of these substances will vary. It will vary depending on several factors. This is one of the very famous questions you will be asked in structure, maybe MCQ also, but mainly in structure. Okay, so this is a structure question. So what are they? The chemical structure and the composition. So very easy for us. If you know, from glucose you will get carbon dioxide, but from proteins you get ammonia. It is because proteins have nitrogen in them, but glucose, they do not have nitrogen in them. They have carbon, oxygen. So they will produce some excretory products according to their chemical structure and the composition. Also, the availability of enzymes. So what are the enzymes that we have that will determine the excretory products? Also, the oxygen availability. How is oxygen availability determining the excretory products? Is because when there is oxygen, it can go under aerobic respiration. If there is no oxygen, it can go under anaerobic respiration. This you know well. And also the habitat. I already told you, the living environment will play a major role in determining these excretory products. We will look at them more later. So these are the 4 factors uh, that will determine the excretory products of these different substrates. So as I already told you, when there is carbohydrate as a metabolic substrate, you have oxygen, it will go under aerobic respiration. So they will be metabolized within the body cells when oxygen is available, and the excretory end products you get is carbon dioxide and water. But if there is no oxygen, it will be subjected to anaerobic respiration, and generally lactic acid will be produced. That you know. Also the fat, same. So when there's oxygen, it will go under aerobic metabolism, and the final excretory gas will be carbon dioxide, water. So now proteins and nucleic acids, they are special. Why? Because they have nitrogen in them. They have nitrogen in them. So proteins contain amino groups. So I already told you how is amino groups converted to uh, ammonia. So during metabolism of those excess amino acids, ammonia is produced. So what is excess amino acids? Some amino acids will be used to synthesize protein. Uh, the excess amino acids only will produce ammonia. Okay. Also, nucleic acids also contain nitrogenous bases. So if you remember the structure of a nucleotide, so this is pentose sugar group. This is the nitrogenous base, and this is the phosphate group. So this guy will be um, turned to ammonia, and that is a very toxic excretory product to our body. So depending on these characteristics, these structures, the excretory products will be varied. Now, this ammonia can be converted to either urea or uric acid. It depends on the habitat as well as the availability of enzymes. So, ammonia can be further converted to these nitrogenous bases. So these are also nitrogenous base. I think uric acid, urea has carbon, so they need carbon for their. They have carbon in their structures. Yes, so ammonia will be converted to urea and uric acid according to the habitat, also the availability of enzymes. So this is what will be moving to the nitrogenous excretory products and the living environment. So we are going to study what is the relationship between the nitrogenous excretory product and the living environment. As I already told you, ammonia is the nitrogenous excretory product that is produced during the metabolism of nucleic acids and proteins. Okay. But depending on the habitat as well as the availability of enzymes, that can be converted further into urea or uric acid. Okay, so these different forms they can vary in their toxicity, also the energy cost of producing them. So to produce urea from ammonia, you need to uh, spend some energy on it. So those energy cost and the toxicity can vary significantly in these different forms. So I already told you ammonia is very highly toxic and it needs large volume of water to excrete ammonia. You need a large volume of water to excrete ammonia. So if you need large volume of water, what is the most convenient uh, living environment? You need more water. So it is better for the organisms that live in water. Okay, so because they have, so if they live in water, obviously they have large volume of water. So typically the organisms that live in the waters like bony fishes, what are the ostic type? And so many aquatic invertebrates. Always remember in A levels, this is not applicable until you reach A level, but when you come to A level, some, those words are very important because this might be a question in your MCQ among 40 to 50. There are ABCD parts where you have to select which are correct or which are incorrect. So they might say, for many, they might give you as some, and if you put it correct, that is a wrong one. Okay, so you have to really consider this many, some, those kind of words in A level. So this many aquatic vertebrates is very important. Not all aquatic vertebrates, but most of the aquatic invertebrates will excrete ammonia, as well as some aquatic amphibians, especially the tadpoles. Tadpole is it is a stage in the life cycle of frog. So it lives in water, right? It really needs water. If there is no water, it will die. So this aquatic amphibian, they can excrete ammonia because they have water, right? They live in water, so obviously they have large amount of water. So they can excrete ammonia as it is. Also, they don't have to produce urea or uric acid. So there is zero energy cost, right? So there is no any further conversion. So the energy cost is very low. Energy cost is very low. Next, the terrestrial animals. terrestrial animals like us, we do not have access to like sufficient water to excrete ammonia. Like we are not living in water, right? Obviously, we are not living in water, so we don't have the access for like sufficient water to excrete a toxic product like ammonia. So therefore, what what happens is the most terrestrial animal like mammals like us and the adult amphibians, they will excrete urea as the main excretory product, main nitrogen excretory product. How how did you get urea from ammonia? Okay, since you don't have access to sufficient water, you decided to excrete ammonia as urea. So you are converting the ammonia to urea and you are excreting it. So urea is less toxic compared to ammonia. So it is less compared. It is less toxic compared to ammonia. Also, now you have to do some work, right? You have to do some work. You have to do a conversion. So you need energy. So you are going to spend like some amount of energy to produce urea from ammonia. Also, some marine fishes like sharks, they excrete urea. That is very important for them in osmoregulation. So that we are not going to discuss here. So, although they have the ready access to water, sufficient water, they chose to excrete urea as their main nitrogenous waste. That is some marine fishes. Remember, some marine fishes. Okay, not all the marine fishes, especially sharks. That is the story of urea. Now, some terrestrial animals like birds and some reptiles, many reptiles, land snails and insects, they will excrete uric acid as the main excretory product. So uric acid, where did we get uric acid from? It is from ammonia. You are going to use some energy to produce uric acid so that you can excrete it using less water. So uric acid is relatively non-toxic comparing to urea and ammonia. It is very, it is very less toxic. Okay, you can even tell non-toxic, and generally it is insoluble in water. So you don't need more water to excrete uric acid. Therefore, it is excreted as semi-solid with a little amount of water, with trace amount of water. So that is why you can see the waste products of birds like semi-solid, but our waste product, what is our urine? That is like water. It is a watery one, but the waste of birds and those reptiles are like semi-solid because of this reason. So what you should really know here is what are the nitrogenous excretory products. Okay, what are the nitrogenous excretory products, and what is the reason for them to vary? You know, two reasons. Always remember them. That is also a very popular question structure. Okay. Then you need to know what is the toxic level. Okay. So yeah, you have to know who is the most toxic person and who is the very less toxic person among the excretory products. Then you need to spend some energy if you need to make one person less toxic. Okay, so you need to know the energy cost and also the examples. This is very important. You need to know all the examples. You can't avoid any one of the example. You need to know everything. So this is the way I study. So I have written a summary of what I've told you until now. So I've compared the toxicity, the energy cost, and the animals. So you also can compare the amount, but that is not very important. If you want, you can compare that one also. So this is the relationship between the nitrogenous excretory products and their living environment. So far, we have finished like what are the metabolic substrate, the excretory products, the nitrogenous excretory products, why they vary, examples, the toxicity, and everything. So this is not very famous for essay questions, but maybe they can ask, because there was one question in FWC asked like to write a short notes on this topic, the relationship between the nitrogenous excretory products and living environment. So what you have to basically write is this. This is the summary. So this is what you have to write. Okay. So far I hope you understood everything. If you have any doubts or any confusions, so if I was not clear anywhere, you can ask me now. and we'll continue. The next, structures. This is the diversity of the excretory structures of animals. This you normally you study in unit 3 also when you were studying the diversity of the kingdom. Himalaya. What is the example? So first is the body surface. That is like you have an order of evolution. Okay? In the order of evolution, you have the body surface first because those animals which use body surface they had direct contact with the environment, so they can eliminate excretory product by diffusion itself. By okay, because they had direct contact with the environment. A very famous example for niditis because from the first itself you might have seen they use this for respiration also, for getting nutrition also. They use body surface for everything. So nidada is the very famous example for that. So next you have flame cells. Flame cells, these are like a specialized ciliated excretory cells which is connected to a network of tubules which opens to the outside of the end. It will be open to the outside of the end. So you can see this is the body wall. So this thing is out. Okay. So you have cilia here. Okay. So this is the structure of flame cell. So this is found in flatworms. Flatworms come under platyhelminths. Yes, platyhelminths. So next you have Nephridia. Nephridia is found in annelids. They are multicellular. Now they are becoming multicellular and they are also tubular structure. One end of the tube is open to the coelom. Okay, will be open to the coelom, and the other end is open to the outside. So here you can see this is the external opening. So this is the coelom, that's the internal opening. So it has two openings, one into the coelom, the other one into the outside. Sometimes they might ask the structure, what is the excretory structure that is open to both, like, into the coelom and outside. They might ask what is the multicellular structure, maybe. Okay, so this is the picture of an epidea. Then Malpighian tubules. So these are the extensive blind end tubules which are immersed in hemolymph. Arthropods they have hemolymph, and it will be opened into the digestive tract. So the examples are the insects and other terrestrial arthropods. Remember terrestrial arthropods. Okay. Next, green glands and internal glands, both are same. So these are the excretory structures found in crustaceans. Who are crustaceans? Do you know any examples for crustaceans?
Shrimp,
Hmm?
shrimp, prawns.
Prawns. Yes. So prawns, crab, they will come under crustaceans. Crustaceans is also a group of. It is a class. It is a class that comes under arthropods. Okay, crustaceans are mostly water living, so they come under arthropods. There are two large glands found ventrally in the head, the ventral part of the head, and which is anterior to the esophagus. So if there is esophagus like this, it will be in the anterior part. Okay, it will be in the anterior part. So you know, right? It's like a common sense, like you already know prawns have their waste matter in the head, so you can remember using that. So the very famous example is prawns, but you can write crustacean. Next, sweat gland. We have the sweat gland because we always sweat during the hot weather. So there are coil tubular glands situated in the dermis. So I hope you studied what is dermis. You will study when you do human skin in sense organs. Yeah. So in the dermis, you have tubular glands, and it is connected to a sweat duct. For example, let's take like this is the will be like tubular structure like this, and it will be open to the skin. So it will be open through a pore on the surface of the skin. So the example is human skin is exclusive to us, I guess. Okay, so sweat glands are exclusive to us. Next is the soul glands. Soul glands, it's why do we need soul glands? To excrete the excess salt. The name itself implies that it is to excrete the excess salts. Sorry, so it is found in marine birds and marine reptiles because they are only mostly related to the salt, right? So uh, they have salt glands and they can excrete using those glands. Okay, so next, the very the very the very famous one and a very important one, the well-developed one is the kidney. So these are the major excretory as well as the osmoregulatory organ. So all the vertebrates, they have this. So this is the major excretory and osmoregulatory organ. Okay, so we have finished like the most boring parts of osmoregulation. Now we are actually entering the interesting part of studying about the human urinary system. Okay, now we are going to start human urinary system. So human urinary system, it's very easy. Okay, it's not complicated like other systems like digestive system or nervous system or something. It's not complicated like it's very simple. It's very crystal clear. You can just look at the picture and tell what are the parts of the urinary system. Now you can see I put a picture here. I didn't write anything because it's very easy to understand by the picture itself. So you can see they have kidneys, two kidneys, and those two from the two kidneys you have the ureters, a pair of ureters from each kidney, and they enter the urinary bladder, and that ends with the urethra. So these are the parts of the urinary system: two kidneys, two ureters, urinary bladder, and urethra. So we need to know what are the main functions of them. So kidney, you already know. It is the osmoregulatory and excretory organ in our body, right? So what is the function of it? Is to produce urine, in which we can excrete the waste product by we can maintain the osmotic balance. But osmotic balance, osmotic balance means like maintaining the balance of solutes. Okay, osmotic balance, osmotic thing is closely related to solutes. Okay, so it is so yeah, maintaining the osmotic balance and the acid base balance. So we'll study how acid base balance later. Next, ureter, it will receive the urine from the kidney. It will receive the urine from the kidney and it will give it to the urinary bladder. And the urinary bladder will temporarily store the urine. And when you have the urge to excrete your urine, then urethra will provide the passage through which the urine can leave the body. Okay, so these are the functions. These are the main functions of the parts of the human urinary system. So it's very easy, right? Also, you can see two important vessels coming in. So the red color one, this is the main blood vessel, aorta. Okay, and this is the inferior vena cava. I hope you know all this already. This is an artery, main artery, and this is the main vein. Vein. Just remember that also. Okay, now we have studied, we have revised what are the parts of the human urinary system. Now we need to, let's just go with a question, okay? Now, if they ask you to explain the total structure of kidneys, you can always start by giving the location of kidney. They ask structure, of course, you don't need to give the location, but it is better to give. Okay, so the location of the kidney you can see here. It is in the posterior abdominal wall. If you take our body and if you touch your abdomen part, you have an anterior part as well as a posterior part. Okay. So if you touch the back of your body, it is the posterior abdominal wall. If you touch the front of your body, that is the anterior abdominal wall. So two kidneys are located on the posterior abdominal wall. Let's take, sorry, this is the posterior abdominal wall. It is on the posterior abdominal wall, and also it is in either side of the vertebral column. So here is the vertebral column. So it is on the either side of the vertebral column, and it is behind the peritoneum. Peritoneum is like a bag. Peritoneum is like a bag in the abdomen, which you really not need to know now. So you will study this later. So it's like a lunch sheet. Okay, it's like a lunch sheet which will cover most of the abdominal organ. It will cover most of the abdominal organ, but it does not cover the kidney. It is behind the peritoneum. So the peritoneum will be here, somewhere here. But it won't cover the kidney. So the kidney is situated behind the peritoneum. Also, it's below the diaphragm. So the diaphragm will be here. You can see the diaphragm will be here. Diaphragm will be there. So this is the location of the kidney. And the very special thing you have to remember, this right kidney is always slightly lower than the left. What is the reason? What do you think is the reason?
Because of liver.
Because of liver, exactly. So you have liver here, somewhere here. Okay. So liver pushes the kidney down. It pushes the kidney down. So when it pushes the kidney down, poor kidney goes down. So the right kidney is slightly lower than the left. Don't forget to mention this with your answer. Okay. Next is the blood supply. Yes, that is the reason why I reminded you of the aorta and the inferior vena cava. So the aorta will bring the oxygenated blood from the aorta. So we'll take this is the aorta. This renal artery here, you can see here. This renal artery will supply the blood to the kidneys. It will supply the blood to the kidneys, and it will be circulated here and undergoes all the filtration process and process urine function, everything, and the the deoxygenated blood will come back into the renal vein. So you know, right? That is a simple thing. Renal vein. So renal vein will be connected to the inferior vena cava. So through the inferior vena cava, it will be taken to the heart. So the blood supply for the kidneys, they receive the blood from the aorta via the renal arteries, and they will return blood through the renal veins to the inferior vena cava. So that is the blood supply. Okay, clear? Okay, I believe it was you all don't have any doubts. If you have any doubts, please ask me. Don't put a message in chat because I won't be able to see. You can turn on your mic and let me know. Okay. Okay. Now we will move to the gross structure of the kidney. What is the gross structure? What is the total structure? We are not going into the deeper part, like not going into the microscopic view of anatomy. Sorry, the kidney. Okay, we are only doing the gross, whatever we can see. You can see a picture here. So this is the external and internal view of a human kidney. This is the external view that you know. This is the internal view. So we are going to mainly study about the internal view here. So you can clearly see it here in this picture. Can you see? Yes. Okay. Yes. Now you might be able to see. So this is a hand-drawn picture. So kidney is a bean-shaped organ. This you all know from grade 2, grade 3. Kidney is a bean-shaped organ which is held in position. Now it should be held in position in the abdominal cavity, right? So it is positioned by a mass of fat, and both are surrounded by, and both of them, I mean, Here is one kidney, there's another kidney also, two kidneys, right? So they both are surrounded by a fibrous connective tissue. There will be a fibrous connective tissue. Fibrous connective tissue around the two kidneys. In the longitudinal section, if you cut it like this, now what have you done? You have cut it like this way. When you cut it like this, you can see three areas of tissues by your naked eye. You don't have to use any microscope to view it. Okay, there are three areas of tissues you can see. They are the outer fibrous capsule. So this is the outer fibrous capsule. Okay, I can draw it completely. So I hope you understand. And then you have the renal cortex. This is the renal cortex. Then you have the renal medulla. And if you study brain, you have medulla and cortex. So there are many cortex and medulla. So please be kind enough to address this cortex as renal cortex and renal medulla. Okay. So these cortex and medulla are supplied with blood vessels and tightly packed with excretory tubules. They are like highly supplied by the blood. So they have a lot of blood vessels and they are tightly packed with excretory tubules. That you know, right? Because those excretory tubules will be open to the pelvis. We'll study what the pelvis is. So this renal cortex, if you see the renal cortex, you can see granules. You can see, right? There are granules. And so if you see the medulla part, you can see some pyramids. So the medulla is completely made up of some pyramids. They're made up of pyramids and they have striated weave. They have a striated weave. Okay, they have striated appearance. It is because the cortex is granulated. The cortex is granulated because it have the glomerulus. Okay, those blood vessels are packed. We'll study what it is in a little while.
Zaidin, you're muted.
Am I audible now?
Yeah.
Okay. So, okay, there are three parts. There are three parts in the longitudinal section of the kidney. The capsule, medulla, and the cortex. So, cortex and medulla, you have to address them as renal cortex and renal medulla. Okay. Renal, you know the cortex is highly granulated. You can see so much of granules here. It is because of the presence of glomeruli. So glomeruli is a network. It is a network of blood vessels. Okay. Then the pyramids, you can see striated appearance. It is because of the loop of Henle that is coming inside the pyramids. That we will study what is loop of Henle in a little while. And then the apex of the pyramids. So this is the apex of the pyramids. They are projected into the renal pelvis. This is the renal pelvis. Okay, through the renal papillae. So these are the renal papillae. Renal papillae. So this is the renal pelvis, and this renal pelvis will lead into ureter. It will lead into the ureter. Okay. Also, you can see the renal artery and the renal vein. They are passing through the pelvis. They are passing through the pelvis. So you should be able to draw this picture if they ask you to explain the gross structure of the kidney. Okay. So you have to draw this. So they will allocate around 6 marks out of 150. Also, you have to label the picture. So, you have to draw and then label the picture. Okay. Okay. Now, we know everything has a structural and functional unit. If we take the whole body of us, the whole living body of us, there is a structural and functional unit. What is the structural and functional unit?
Nephron. Nephron.
Really sorry for the interruption. So, as I was saying, the structural and functional unit of the kidney is the nephron. Okay, so this is the picture of a nephron. So then the nephron is the one who is going to do all the important functions of the kidney. So there are over millions of nephrons in each of the kidneys. Okay, each of the kidneys. So there are mainly two types of nephrons. Okay, there are two types of nephrons. We call them cortical nephrons and then juxtamedullary nephrons. So you can see in this picture, this is a cortical nephron and this is a juxtamedullary nephron. The cortical nephron, they reach short distance to the medulla. So this is the cortex and this is the medulla part. So in the cortex, from the loop of Henle is reaching a little distance in the medulla. That is the cortical nephron. But the juxtamedullary nephrons will reach to a deeper distance into the medulla. So this is the very primary difference between the cortical nephron and juxtamedullary nephrons. So it is said that cortical nephrons are of 85% in each kidney and the juxtamedullary nephron is like 15%. Okay. The difference is the loop of Henle will extend deep into the medulla and the cortical nephron's loop of Henle will reach short distance to the medulla. That is the difference between the cortical nephrons and juxtamedullary nephrons. So as I told, the majority of the nephrons are cortical nephrons. Okay. Now let's uh, see about the nephron. Okay. So a nephron, it is a single long tube. Okay, it is a single long tube, but since it is a small one, we call it as tubule. Okay, a tube when it is small, we call it tubule. So nephron is a single long tubule. So you can see it starts from here, from this to you can see the blue color part. That is blue color part is the single long tubule of a nephron. So the tubule is closed at end, forming the Bowman's capsule, which will surround the ball of capillaries called the glomerulus. Glomerulus. So this is the ball of capillaries I was talking earlier. This is why the cortex was granulated. It looks granulated because of the glomerulus there. Okay, so it is a ball of capillaries. The capillaries will be there as a ball. Y'all can hear me, right?
Yes.
Give me a minute. Okay, let's continue. So an nephron is the um, in one. So you can see this is one end. So this is Bowman capsule. Okay, this is the Bowman capsule. It will surround the ball of capillaries. It will surround the ball of capillaries, which is called the glomerulus. Okay. Um, this is called the glomerulus. So this is the reason why the cortex was granulated. The other end will be joined to the collecting duct. Now tell me, is the collecting duct a part of nephron? I am expecting an answer from no. Yes. Collecting duct is not a part of nephron. Okay, remember that. It is not a part of nephron. So nephron finishes at distal convoluted tubule. Okay. So one end will be the Bowman's capsule, which surrounds the ball of capillaries, and the other end will be joined with the collecting duct. So this tubule will have mainly four parts. The first one is the Bowman's capsule. Then you have the proximal convoluted tubule. Then the loop of Henle. This loop of Henle we can further divide into parts that I will tell later. And then it will continue as distal convoluted tubule, which will ultimately reach the collecting duct. Okay. So first, the Bowman's capsule. So Bowman capsule, we can also call it as glomerular capsule because it surrounds the glomerulus. So this is an expanded and closed end. Right? It is a closed end. It is closed here. But here it is an open end. Here it is an open end. The other end is an open end. But this is a closed end and it is expanded. You can see like here it is kind of expanded. And Okay, so inner layer of the Bowman capsule will consist of a single layer of flattened epithelial cells specialized for filtration. If there is filtration, then it is no use if if the wall is such, right. So the inner layer of the Bowman capsule will having only a single layer of the flattened epithelial cells for filtration. The outer layer of the Bowman's capsule will be composed of simple squamous epithelium. I hope you know everything about the epithelium since you might have studied in the very beginning of this unit. So it will have simple squamous epithelium. Simple squamous epithelium is very thin. So therefore filtration can take place very easily. So the space between the inner and outer layer, the space between the inner and outer layer, how do you call it? Is the capsular space which will be responsible for receiving the glomerular filtrate. We will see what is glomerular filtrate later. So this glomerular filtrate will pass through the major three tubules. What are they? The proximal convolutor, the loop of Henle, and the distal convolutor tubule. So this is pretty much about the Bowman's capsule. So you know it is a closed and expanded cup shape, double walled, the inner layer and the outer layer. Inner layer is of the single layer of flattened epithelial cell, and the outer layer is of the squamous epithelium, and there is a interspace between the two layers, that is capsular space, and it is responsible for receiving the glomerular filtrate. Then this glomerular filtrate will continue its journey through the major tubular regions of the nephron. They are the PCD, Henle, and the DCT. Okay. Next, we have the glomerulus. So, as I already told, glomerulus is a bowl of capillaries. It is a bowl of capillaries which is surrounded by the Bowman's capsule. You can see the Bowman capsule has surrounded the glomerulus. So, the nephron is supplied with blood from the efferent arteriole. Now, I told you from where the kidney receives the blood from. It is from the aorta, right? From the aorta, from the renal artery, the renal artery, it will give so many branches. One of the branch is the efferent arteriole to the one nephron. So it will give efferent arteriole to many nephrons. So one nephron. So the blood will enter efferent arteriole. It will enter the efferent arteriole and it will go through this capillary, go through this network of capillary. When it is going through the capillary, the blood will be filtrated. Those uh, those molecules which are not needed to our body and which are very small, they will be filtered into the Bowman's capsule. That filtrate is what we call the glomerular filtrate. It is coming from the glomerular, so it is glomerular, and it is a filtrate, so therefore glomerular filtrate. It will move here and there, and it will just go through it, and it will then come back out through the efferent arteriole. Sorry, the efferent arteriole, and it will go back to the capillary network like here, and it will move here, go somewhere here, and it will finally come here, and it will go out and reach the venule, then the renal vein, and it will reach the inferior vena cava. So this is the part of the blood. So nephron will receive the blood from the efferent arteriole. The blood vessel that will leave the glomerulus is the efferent arteriole. So here there is something special. You can see that the diameter of the efferent arteriole is smaller than the diameter of the efferent arteriole. It is because uh, the blood pressure is increased in the glomerulus for the ultrafiltration. If there is a high blood pressure, then it will be good for the filtration. Therefore, that is why this adaptation is there. What is the adaptation? The efferent arteriole has a smaller diameter than the efferent arteriole to increase the blood pressure in the glomerulus. That is for the ultrafiltration. Okay. So two capillary networks, okay, that we have to see. It forms two capillary networks around the tubule, right? So this is one of the capillary network. This is one of the capillary network. Okay. Uh, One form is the peritubular capillaries that will surround the PCT and DCT, the proximal convoluted tubule and distal convoluted tubule. The capillary network that surrounds these two parts, we call it as peritubular capillaries. Peritubular capillaries. And the other network that will surround the loop of Henle here is the vasa recta. Vasa recta. So these are the two types of capillary networks that are formed by the different arteriole. The peritubular capillaries will surround the proximal and distal convoluted tubule, and the vasa recta will extend towards the medulla surrounding the loop of Henle. Okay, so this is pretty much about the glomerulus. Next, we have to move to proximal convoluted tubule. So proximal convoluted tubule is next to the Bowman's capsule. So the Bowman's capsule, and it is comparatively longer and wider. It is longer and wider than the distal convoluted tubule. Okay, it is like longer and wider. Also, this is lined by the simple epithelium because during the process of urine formation, we will study about selective reabsorption and secretion. For selective reabsorption as well as secretion, you need to have a thin wall. If there is a thick wall in between the two region where there is selective reabsorption and secretion, it will be very hard. So you should have a thin wall. That is why this is lined by simple epithelium, which is specialized for selective reabsorption of substances from the glomerular filtrate. So that is about proximal convoluted. That's it. There's nothing else about the convoluted tubule. Next, you have the loop of Henle. When you see the picture itself, you can say it is a U-shaped part of a nephron. And I told it has two parts. What are they? This is the ascending limb. This is the descending limb because it is coming down, right? Descending limb. And this is the ascending limb. Ascending limb. Right? So they are also lined by simple epithelium. I already told the reason. That reason will apply to this as well as to the distal convoluted tubule. Lining of the so it is lined by the simple epithelium. And there is another special thing: the lining of the descending limb. This limb is specialized for water reabsorption. It is a specialized one for the water reabsorption, and so it will allow the free movement of water. But in contrary, the ascending limb will be impermeable to water. It won't allow a single molecule of water. But the descending limb will allow all the free movement of water. But the descending limb won't. So that is a specialty of the loop of Henle, where descending limb and ascending limb are working opposite to each other in water reabsorption. So that is all about the loop of Henle. Then you have the distal convoluted tubule. So here you can write add the sentence like it is shorter and narrower than the proximal convoluted tubule. The same one is simple epithelium. Why? Because for the selective reabsorption, also it will be the one who will lead us to the collecting duct. So distal convoluted tubule is the one who is going to lead us into the collecting duct. So this is the structure of nephron. So you can get a question, explain the gross structure of kidney, or I'll explain the structure of a nephron. So if they ask question, you should be able to explain of this nephron. Okay, don't forget to draw. You will get 6 marks, and don't forget to label. If you just draw and you didn't label it, you won't get any marks. So don't forget to label it. So that is all about the gross structure of the kidney as well as the nephron. So next we are moving into the urine formation. If you have any doubts, you can ask. So I hope no doubts. we will move to the process of urine formation. In the process of urine formation, you have three steps, three main steps of urine formation. What are they? They are the ultrafiltration, selective reabsorption, and excretion. Okay, so these three process, they can ask you in structure what are the name the three process involved in urine formation, or name the main. Okay, so those are the questions that can be asked under this topic. Essay question is most preferably asked under this topic because MCQ structures they can take, but also essay is very prominent here. So first, let's go through all the three process. First is the ultrafiltration. So when you write answers, you have to remember one thing. uh, Take it for ultrafiltration or selective reabsorption or secretion. The first thing you have to write, that is the definition. The first thing you have to write the definition. So for ultrafiltration, what is ultrafiltration? It is the filtration of the blood. Okay, now you have the efferent arteriole here. Okay, efferent arteriole, and it will be like this is the glomerulus, something like this. And you have the efferent arteriole. This is the Bowman's capsule. The blood will come, and there is high pressure here, and under the high pressure, some will be filtered. Okay, some will be filtered. So under the high pressure, it will be filtered into the cavity of Bowman's capsule. So it's very easy. Filtration of the blood under the high pressure into the cavity of Bowman's capsule. That is what you call ultrafiltration. So filtration occurs through the capillary walls of glomerulus. Okay, this is the capillary wall of the glomerulus. It will happen through it, and then the inner wall of the Bowman's capsule. This is the inner wall of the Bowman's capsule. Okay, so the filtration will go through it. So the blood capillaries of the glomerulus, they are porous. Okay, the blood capillaries, they are porous, like very small pores will be there. And the cells lining the Bowman's capsule, these cells lining the Bowman's capsule, they have special cells called photocytes. Okay, so they can allow the movement of some small molecules inside the Bowman's capsule. So blood capillaries, they are anyway porous, and the cells lining of the Bowman capsules are specialized for filtration of what? The small size molecules and ion. It won't let go through of the big molecules because it is very tiny one. So this specialization will allow passage of water and small solids through the blood capillary walls into the Bowman capsule. This is how the water and small solids are going into the Bowman's capsule. The large molecules, they can't pass through these walls because they are large. Obviously, they are large. So what are they? Blood cells. Blood cells will include RBC, WBC. If RBC is if RBC is filtered through this wall, our urine will be in red color. Okay, but they are large in size, so therefore they won't be able to pass into the Bowman's capsule. And also you have platelets and the plasma protein. What are the plasma proteins? Like albumin, fibrinogen. They won't be passed into the Bowman's capsule. Also, they are very important to our body. We can't let them just go. Okay. So this filtrate in the Bowman's capsule. This filtrate, now there is something here, right? We had something here. It will contain like water, some ions, amino acids, glucose, vitamins, some nitrogenous waste, some molecules. What is the problem here? We need amino acids, we need glucose, we need vitamin. Why did we eat if we don't need it? Okay, we need them back. So what we should do? We have to regain them back. So we have to reabsorb them. So that's what going to happen in the second step. So they are unfortunately filtered because they are small particles. They are small particles. Also there was high pressure there. So it was filtered. But our body really needs amino acid, glucose, and vitamin. That is the whole reason why we eat, right? So if we just let them go through urine, it is no use. It is of no use. So therefore, we have to reabsorb them back. Okay, so this is what going to happen next. Also remember the composition of the glomerular filtrate. This filtrate that will be similar to the composition of plasma without plasma proteins. Without plasma proteins, okay, they will be equal to the plasma, similar to the plasma, but the plasma proteins will be not there. Sometimes they might ask you in what MCQ. Okay? They might ask you in MCQ if it is they might give us a statement like the glomerular filtrate is similar to the composition of plasma. If they just say like that, it is false. If they say without plasma proteins, that is true. So now let's move to the second step. So you already know the importance of means we need minerals, we need glucose and some ions. Also, we need water to our body. We can't lose all the water out. So we have to reabsorb them. So the process where the useful molecules, ions, also water from the glomerular filtrate. So now this is the Bowman's capsule. So the filtrate is here now. The filtrate is here now. And you have the tubules. I'm just like rough, it's a rough drawing. So here is the filtrate. Now we have to get whatever we need back. So what happens is the useful molecules, iron, water from the glomerular filtrate will be recovered and will be returned to the interstitial fluid. So this is the around the nephron we have the interstitial fluids. So it will be moved to the interstitial fluids, and we told there are two capillary networks around the PCT, DCT, and the loop of Henle. Remember the peritubular network and the vasa recta. So here we will have the peritubular network. So therefore, uh, those were reabsorbed into the interstitial fluid will be entered into the black back. So they will again go into the inferior vena cava through the way I told you earlier. Okay, so it's like this. Now look at this picture. So here now it is from the DCT, sorry, from the PCT, the proximal convoluted tubule, we have reabsorbed some glucose. Let's take for example. So now that is in the interstitial fluid. From the interstitial fluid, it will be taken into this peritubular network. Then it will move like this. Then it will like just go through this and go through this, and enter this one, and then enter the venule, then vein, and inferior vena cava. Now it is back into the systemic circulation. Okay, so that is the main purpose of selective reabsorption. Okay, most of the reabsorption of the glomerular filtrate will be taken place in the convoluted tubules. Okay, more than the loop of Henle, it will be happen in the convoluted tubules. The ions are like ions like sodium, water, and some valuable nutrients. What are they? Glucose, amino acids, vitamins. They will be reabsorbed either active or passive. There will be two types of transport. Maybe active using ATP or the energy, or passive without using energy or ATP. Okay, from the initial filtrate. So next is the secretion. Why secretion? So now I already told during the ultrafiltration there will be high pressure, right? High pressure. So there will be filtration here, there will be filtration here, but the blood stays in the capillary network for a very few, like few seconds. Okay, few, the time is very less. So therefore, not everything that we don't need will be filtered. Some toxic things that are not needed for our body will not be filtered because of the less time. So therefore, what happens is the secretion comes to play. So what the now let's take ammonia. Ammonia, let's take like some toxin from our body is not it's not filtered. It is not filtered into the filter. So what happened is the secretion during the secretion it will be secreted by how? Look at this picture again. Whatever not filtered will go through this way, right? It will come like this and it will come through this. And here it will be secreted into the interstitial fluid and then it will come back into the PCT and then it will go. This will happen in the proximal convoluted tubule as well as the distal convoluted tubule. Like sometimes in the main parts are proximal convoluted tubule and the distal convoluted tubule. This is secretion. So the process by which the foreign material and substances that are not required to our body, including the waste, will be cleared from the peritubular capillaries and the interstitial fluid. It will into the filter. That is what we call the excretion. Okay. So the secretion is required because substances may not be entirely filled because of the short time they remain in the glomerulus that I already told. So the substances like H plus, in ammonia, creatine, it is also a nitrogenous phase product. Remember, this is a nitrogenous phase product. Drugs like penicillin, aspirin, and excess K plus. Always remember to tell excess. Don't just say K plus. Say excess K plus. Okay. And also they will be secreted. Now the H plus and NH3 will be secreted into the tubules and they will form NH4 plus. So you know NH4 plus is a buffer. So it will maintain the normal pH in the blood. Okay, it will maintain the normal pH in the blood by combining H plus with the NH3. So secretion also will be happening both the convoluted tubules and it may be either active or passive depending on their location and the substance. Okay, this is the steps of urine formation. Now, if they ask you to explain the process of urine formation, you have to say what are secreted, what are reabsorbed, what is passive, what is active, if it is diluted or if it is concentrated. These are and the other thing is about the buffer. These are the main things you have to write when they ask to write you about the process of urine formation. They ask the steps of urine formation. You have to write whatever we studied until now. Now we are going to study the process of the urine formation. For that, we will take this picture. Not this one. This one. So this I have tried to summarize. So this is in your resource book also, but I have given different colors and things. You can see. Now let's start from here. Now it is now ultra filtered. Now the glomerular filtrate in the Bowman's capsule, it will contain all the substances in the blood except the blood cells, platelets, and the large molecules, right? Like plasma, protein. The others will be moved to the proximal convoluted tubule. In this region, selective reabsorption will happen. So there is selective reabsorption of ions like sodium, water, and some valuable nutrients like glucose and amino acids from the initial filtrate will occur. Especially, we need to get all the nutrients back into the body. So glucose and amino acids, right? Generally the nutrients they will be reabsorbed actively. So I have written in red color. They will be absorbed actively. Actively they will be transported in the interstitial fluid, and you know what will happen after interstitial fluid. Now the cell lining, the tubule actively transport Na plus. So Na plus is also transported actively into the interstitial, and since the positive ions have gone out, Cl minus is the best friend of Na plus. So he will always follow Na plus wherever the Na plus goes. Okay, it's like a test. of no, like a best friend of Na plus. Since it is plus, sodium ion is plus and chlorine is minus, it will go behind. So it is like the passive transfer. So you can see it is a passive transfer. I have written in black color. Okay. So proximal tubule will also reabsorb some K plus. Okay, some K plus, but that is passive. And most of the bicarbonate ions by the passive transfer. Now you know bicarbonate ion is one of the important buffer, right? So he will, what will it do is it will contribute to the pH balance in the body fluid. So it will contribute to the pH balance. So wherever the buffer comes, don't forget to mention that it will contribute to the pH balance. Just tell it will contribute to the pH balance and your essay will be complete. So now the solutes will move from the filtrate to the interstitial fluid. The water will be reabsorbed passively by the osmosis. So wherever the water will be reabsorbed passively, okay, through the process called osmosis. So the major portion of water will be reabsorbed from this proximal convoluted tubule, and the secretion of specific substances also will take place here. What are they? The cell lining of the tubule will secrete the H plus. So H plus will be transported actively. So that is why I've given a different color. Also ammonia by passive transport. It will be secreted by passive transport into the lumen of the tubule. Next, secreted ammonia can form NH4 plus with the H plus, and it can buffer again. So you can write it is a buffer. In addition to that, some drugs and toxins that have been metabolized in the liver, they will be actively secreted into the proximal convoluted tubule. Okay, so as a result of water reabsorption and so many secretion of different substances, what happens? There is less water inside the proximal convoluted tubule. So the filtrate is more concentrated there. So you have to, so it's very easy, right? You have to remember like what are reabsorbed, what are secreted, either it is passive or active. Tell if there is any buffer and tell if it is concentrated or diluted. That's all. You have to follow this for each. Okay. Next, as the filtrate moves into the descending limb of the loop of Henle, passive reabsorption of water will take place. We already studied that descending limb is adapted for the water reabsorption. So it will allow water to be reabsorbed through the osmosis. And the filtrate will be more concentrated, right? Because the water is again reabsorbed. Now the filtrate will reach the ascending limb of the loop of Henle, and the ascending limb is impermeable to water. Okay, so there's no any water reabsorption takes place, but there is a considerable amount of NaCl reabsorption that will take place, reabsorb actively as well as passively. That is a different thing. So that's what you have. That's what you have in your resource book. So you just remember as it is, but that has a different story also. That it is really unnecessary. Just remember whatever is in your resource book. So most of the Na plus is reabsorbed into the interstitial fluid by the active transfer that you might have known by now. So as a result of losing NaCl and no any water reabsorption, so there will be some water inside the ascending loop, right? So therefore, this is one of the only eluted. Then it will move into the distal convoluted tubule. Again, distal convoluted tubule will play a very major role in regulating the K plus and Na plus concentration of body fluids. Can one of you say why is K plus and Na plus so important to our body?
They are important in activating signals of the nervous
Yes,
system.
exactly. So it is very important for the nerve impulse transmission, right? So yes, that's the thing. So K plus and Na plus is very important for us to regulate them. So the amount of K plus will be is secreted into the filtrate, and the amount of Na plus that is reabsorbed by the filtrate will be varied according to the needs of our body. So K plus secretion as well as Na plus reabsorption both are by active transport. Okay, they both are by the active transport. So here it is like it's so boring. Just I have to just say that is secreted, that is active, this is reabsorbed, that is passive. You just have to remember. There's no other way. Okay, so the distal tubule will also contribute to the pH regulation by some secretion of H plus there, also absorption of HCO3 minus. They haven't told if it is active or passive, but in your picture it was passive. In the picture they have given in your resource book, I found it is passive, so that's why I have given the color of passive. Active. Active secretion and active reabsorption of HO3 minus. So that is active. Okay. So in the distal convoluted tubule, there is a special thing that happens, that the passive water reabsorption will be further increased by a hormone called antidiuretic hormone, also which is commonly known as the ADH, to form concentrated ions. So it will result in water reabsorption, passive water reabsorption. Also, there is another hormone called aldosterone, which will be secreted by the adrenal glands. We will study more about ADH and aldosterone. We study homeostasis or hormonal coordination. Okay, so here you just have to remember what are the. So, do any of you know which part of the body secretes ADH? Which secretes ADH?
Posterior pituitary
Posterior?
hormone.
Yes, yes, exactly. Posterior pituitary gland. So it will produce, sorry, it will secrete two hormones, ADH and oxytocin. So ADH is from the pituitary gland, so it will come from the brain to the kidney. It will travel a long journey. Okay. So aldosterone, which is secreted by the adrenal gland, it will stimulate the reabsorption of Na plus as well as water. Also the excretion of K plus. So K plus. So this filtrate of the distal convoluted tubule will finally lead into the collecting duct. So aldosterone will mainly work on the collecting tube, okay, collecting duct. So as the filtrate passes along the collecting duct, filtrate will become more concentration and the urine here. This is where you call urine, okay. The collecting duct is where you call urine. Until then, you called it glomerular filtrate. There's the reabsorption of Na plus and passive reabsorption of water at the collecting tube. Also, there will be a high urea concentration in the filtrate. Therefore, urea will be diffused into the interstitial fluid. Finally, we have the concentrated urine in the collecting duct. This collecting duct will go into the pelvis and it will go into the ureter. Okay, so when producing, if we need to produce more dilute urine, the kidney will actively reabsorb the salt and without allowing the water to be reabsorbed by osmosis. So the water reabsorption will be very less. Then we'll have the dilute urine. It depends on the situation. So this is all about the process of urine formation. So there's a lot to remember, but if you remember, that is very simple, right? So you don't have like remember word by word. You can build up the answer for your question. So this is like you can make a chart like this. If you can remember like this, then you can draw it like this and remember. Or else if it is easy for you to remember like this, you can put a chart like this and remember. Okay? It is up to you. Okay, now we are going to move into the last part of this discussion. So, do you have any doubts until now?
At the ascending group of Henle, you have marked at the ascending group of Henle.
Sorry.
At the ascending limb of Henle,
Yeah, yes.
you have marked NaCl as both passive and active.
Yes, that is what mentioned in your resource book. That's what I told you. But that is really a different case. Okay, that is not actually true. It is reabsorbed passively and uh, actively. It is not really true. But that is what they have given in your resource book. Like NaCl reabsorption will occur actively as well as passively. That's all. But the real case is different. Okay. But just remember
Ah,
what is in your book. Okay.
okay, thank you.
Sometimes your biology teachers might have told what is really there and something. I don't want to complicate, so you can just remember what is in your book. Okay, so now let's move to the final part, that is the role of hormone. This is the most interesting part of the biology. Like, actually, you can work it out. So first, we'll see what is the role of hormones in regulation of blood osmotic pressure and fluid retention in the kidney by the ADH. Okay, so blood osmotic pressure means the amount of solutes, the concentration of solutes in the blood, right? So if there is increased blood osmotic pressure, that means what? The blood the water level, it will be sensed by the osmoreceptors in the hypothalamus. So where is hypothalamus? It is the brain. It's a part of the brain, right? So hypothalamus, in the hypothalamus you have osmoreceptors. Osmoreceptors are one of the receptors that will sense the blood osmotic pressure. When there is increased blood osmotic pressure, osmoreceptor will sense it. So it will stimulate the posterior pituitary gland. Okay, so it will stimulate the posterior pituitary gland. Also, the hypothalamus will generate the thirst. So you will drink more water. Okay, that is one case. Then, when stimulating the posterior pituitary gland, the ADH will be secreted into the bloodstream, and it will travel along the bloodstream, and it will reach the kidney, and it will work on the distal convoluted tubule as well as the collecting duct, and it will increase the reabsorption of water. So, therefore, the water level in the blood increases and the osmotic pressure in the blood reduces. Okay, that is how the blood osmotic pressure returns to normal. When it is returned to normal, it will inhibit the osmoreceptors in the hypothalamus, preventing further reabsorption of water. So this is the regulation of blood osmotic pressure uh, by ADH, which is done by the kidney. Okay. The second thing is the regulation of blood volume and blood pressure by renin-angiotensin aldosterone system. We usually call it as RAS system. The first letters of renin, angiotensin, aldosterone system. Okay. So here we have to start from here. The blood pressure or blood volume can be low. Okay, so the blood volume will determine the blood pressure. If the blood volume is high, then the blood pressure will be high. So if there is a drop in the blood pressure or blood volume due to like dehydration or blood loss during accident, hemorrhages like or severe burns, there will be loss of blood or water. Okay, so therefore the blood pressure or blood volume in our body will drop. This will be sensed by the kidney. In kidney, we have a special system called juxtaglomerular apparatus. This is not in your book, but just remember because there are questions regarding this. They might ask what is the sensor that detects the decrease in blood pressure or volume. If you have to answer that juxtaglomerular apparatus, there are actually questions on this. So remember. So this person will sense there is a decrease in blood pressure or volume and it will secrete renin. Renin is a type of enzyme. So this renin, which is an enzyme, will be secreted by the kidney. So this renin will convert angiotensin also, which is produced by the liver Okay, so the renin will be secreted by kidney, and it will convert angiotensinogen to angiotensin 1. So angiotensinogen is from the liver. This angiotensin 1 will be converted to angiotensin 2 by angiotensin converting enzyme. Angiotensin 2 has so many effects, but here we are going to see only two. One is it will constrict the arterioles. When the arterioles are constrict, it will help in the increasing blood pressure by so many things happening after that. But you don't need to know. Okay, so when the arteriole constrict, there will be an increase in blood pressure. Also, angiotensin 2 will stimulate the adrenal gland, and the adrenal gland will secrete aldosterone into the bloodstream. So this aldosterone will now come to the distal tubule, and more sodium and water will be reabsorbed in the distal tubule, which will increase the blood volume and blood pressure. So now the normal blood pressure and volume is maintained because of aldosterone. So these are the two regulations we study here. These are the two regulations we study here. So here you have when the blood pressure or blood volume drops, you also have to write what happens when the blood pressure or blood volume increases. Like you can write one or two sentences, which is vice versa to this. Okay. Same applies to this one. Okay, so this is like a very easy essay. If you get this in essay, that is very easy to score marks. So this is all about today's discussion. There's another one more part, but I didn't concentrate much on the disorders, because if you read and memorize, that's all about it. um, you can ask if you have any doubts, because we are like, in the end of the session. Just remember the juxtaglomerular apparatus. Okay, that is not in your book, but there are questions regarding that. Also, renin is an enzyme. Sometimes you might get an MCQ like renin is an hormone, then that is wrong. Renin is an enzyme. Do you have any doubts? Okay, I hope there are no any doubts. So we'll do a quick summary of what we did today. So we were going through osmoregulation and excretion. So we studied what is the importance of osmoregulation, excretion, the definition of osmoregulation and excretion. Then we studied about the importance and need of it, and why we need osmoregulation as well as the excretion. Then we studied about the metabolic substrate and what are the excretory products from them, and are the factors that determine the ammonia according to the habitat and availability of enzyme, and we compared the toxicity, energy cost, and the animals that are excreting these excretory products. And then we move to the excretory structures that have been developed for the evolution of the kingdom animalia. So we went through some examples, and then we moved into human urinary system, the main parts of the urinary system and the functions of each of the system. Then we studied about the gross structure of the kidney, the location, the blood supply, and the gross structure of the kidney. You have to write them. Then we studied about the functional and structural unit of the kidney, which is nephron, which is a long tubule with the glomerulus. So the long tubule has Bowman capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule. Collecting duct is not a part of nephron. Okay, so there are two types of nephrons, the cortical nephrons and juxtamedullary nephrons. So we saw what are the differences between the cortical and juxtamedullary nephron. Then we had the process of urine formation. In that, we studied the main step, ultrafiltration, selective reabsorption, and secretion. You have to write the definition first, and what are the adaptation and why it is happening, and what is secreted and reabsorbed passively and actively. Then we studied the process of urine formation actually. So what happens in each part of the tubule? What is secreted? What basically what you should write, what happens to the glomerular filter, what is reabsorbed, what is secreted, either it is passive or active. Then you have to write if it is a buffer or not, so that it helps in the pH balance or acid base balance. And then you have to mention if it is concentrated or diluted filtrate. That's it. Then you have to talk about the ADH as well as ADH and aldosterone in different parts. Then you have two regulations done by the kidney. One is regulating the blood osmotic pressure, and the other one is the blood pressure and volume. So those are the main things we discussed today. So if you have any doubts, you can ask me. You can text, put a text in the chat also. You have I really didn't get your questions, the question like writing about the component reabsorption when they ask about glomerular filtrate in it. There are examples for marine birds, but seal, right? Seal, seal, I think so. No need to bother about that, you can just write marine birds. To remember the essay points, just make some flow charts and remember. Anything else? RAS mean that is the renin angiotensin aldosterone system. That is the system where the renin was secreted, and it turns angiotensinogen to angiotensin 1, and then angiotensin 2 turn to angiotensin 2. That regulation, that is RAS. Okay. Then don't to write about each you have to write. They will ask question. There will be what are the main. They will ask what are the main steps. Explain the main steps of process of urine formation. Okay. They ask you have to write those 3 steps. Okay. Sometimes they will ask write the process of urine formation. Process of urine formation. When they ask like that, you have to write about everything, the selective reabsorption as well as the okay. Anything else? Can you please tell me the most important lesson? 1 to 5 units are very important. 1 to 5, be completely clear with 1 to 5 units. Okay, those first 5 units are very important. The most important lesson will be the unit 5, because you will get like 2 essays from 5th unit. Maintaining homeostasis. Okay, inshallah, we'll try to. Then 5th unit, fifth unit is the main one, and also study the 2nd unit completely. 2nd unit, 5th unit, 4th, very important. But for MCQs and structures, obviously you have to study that all 10 units, because you might be getting like, for the structured essay is different. Like the first question is based on unit 1 and 2. Second question is based on 4, 5. Third question is based on uh, molecular biology, genetics, that. And the fourth question will be based on the applied biology and the microbiology. So obviously you have to study everything for MCQs and structure. But if for essays, if you are targeting for essays, maybe 2nd unit, 4th and 5th. If you study those 3 units for essay, obviously you can like choose all the, but you have to be thorough with the 3 units. Anyway, try to study all the 10 units. Anything else? Okay, then we will finish. Thank you so much for joining.